Hi, today we are going to look into our next topic, transport system in plants. We all know that we need food and oxygen to live. Plants also need energy or glucose to grow. Photosynthesis in plants needs sunlight, carbon dioxide and water to produce energy and oxygen. Do you know how the leaves at the top of a tall tree obtain water that is necessary for photosynthesis? Yes. Water is transported in the plants via the vascular bundles which are located in the roots, stems, and leaves. This means that plants also possess their own unique transport system. Therefore, we will look in detail at the vascular tissues which consist of the xylem and phloem, which make up the plant's transport system. So let's now look in detail at the transport system in plants. There are two types of tissues involved in the transport system of plants. These are the xylem and phloem, and they are also known as vascular bundles. And they are found in the roots, stems, and leaves. What is xylem and phloem, and what are their functions? The xylem is a dead cell without a nucleus. The xylem transports water and minerals from the soil through the root system up the stem to the leaves. Xylem vessels are supporting tissues for the plants and they form the woody part of the stem. Whereas phloem tubes are alive and they transport the glucose produced by the leaves during photosynthesis to all parts of the plant. If we depend on our heart as the main organ for the transport system in humans, then plants depend on the xylem and the phloem for their transport system. Let's examine the xylem in the first experiment to see how water and minerals obtained from the soil are being transported through the stem and leaf.
For this experiment, we need a beaker of water, conical flask, eosine powder, spatula, blade, microscope, glass slide, cover slip, a balsam plant, and celery stem. First, we need to fill half the conical flask with some water. Then, take a spatula of eosine powder and mix it with the water. The water is now red in color. Then, take the celery stem and place it inside the conical flask. And now, repeat the same procedure but using the balsam plant. After 24 hours, let's see the result for ourselves. Now, take out the celery stem and place it on a dissecting tray. Cut a thin slice of the stem and place it on the slide. Place the cover slip on it and look at it under the microscope. Examine it closely. Now, Through this observation, we can see, based on the cross-section of the celery stem, that the xylem tissues, which are arranged in rings in each vascular bundle, are also red in color. We can also observe that in the case of the balsam plant, the red-colored water has traveled up the roots through the stem to the leaves. This enables the leaves to carry out photosynthesis. Therefore, from this experiment, we can conclude that water from the roots is transported to the leaves in the xylem tissues. In this experiment, we will now examine how plants transport the food they produce during photosynthesis. For this, we need a blade and a woody plant, such as pudding plant. We need to cut a complete ring of the bark from the stem, including the phloem, to expose the xylem. Make sure that the phloem is completely removed. Then, leave the plant aside for 2 to 3 weeks and observe the effects after this period. We can now see that after 2 to 3 weeks, the bark above the ring swells. This is because food synthesized in the leaves cannot be transported below the ring because the phloem had already been removed. Therefore, food accumulates just above the ring and causes the bark to swell, whereas the bark below the ring shrivels because food below the ring has been used up. Since food from the leaves cannot be passed down, the cells of the bark die and finally shrivel. We also observe that the leaves on the upper part of the ring do not wilt throughout this experiment because water can still be transported up through the xylem tissue that was left intact. Therefore, we can conclude that phloem in the bark plays an important role in the transport of food from the leaves to other parts of the plant.
Transpiration is the process by which plants release water into the air through tiny pores or openings on the leaves called stomata. Stomata can be found more on the lower epidermis of the leaf compared to the upper epidermis. Stomata have a pair of bean-shaped gut cells to control the loss of water from plants. These cells control the pore size of the stomata. Besides water, gases like oxygen and carbon dioxide diffuse in and out of the leaf through the stomata during photosynthesis and respiration.
In this experiment, we will identify the stomata and the surrounding cells where transpiration takes place. For this, we need to use a microscope, slide, cover slip, scissors, forceps, nail varnish, and leaf. OK. Take the leaf, use the scissor and cut a section. Now, take the nail varnish and coat the lower epidermis of the leaf and let it dry for a few minutes. Next, use the forceps and peel off the varnish layer slowly. Place it on a slide. Use a cover slip to cover the layer. Let's examine it under the microscope using low power to identify the stomata. We can now see clearly the epidermis cells with the stomata and its surrounding guard cells. The stomata of the plants are normally open during the day for photosynthesis to occur. This allows for the exchange of carbon dioxide and oxygen. At the same time, transpiration, which results in loss of water, occurs through the open stomata. On a hot day, the stomata will close to reduce water loss when the rate of transpiration exceeds the rate of water absorption. Now, we will study the factors affecting the rate of transpiration of the upper and the lower surfaces of the leaves. For this, we need 5 balsam plants, conical flask, beaker of water, oil, cotton wool, and a triple beam balance. And this experiment here is to study the rate of transpiration by using 4 manipulated variables, one at a time. Now, take a conical flask and fill up with 180 ml of water. Then, choose a healthy young balsam plant. Put the plant inside the conical flask and pour a layer of oil over the water surface. Next, cover the mouth of the flask with cotton wool. Weigh the conical flask and its contents. Leave the apparatus in the lab under normal room temperature as control set. Repeat the same procedure again. But now, we put the second apparatus under the fan. After 2 to 3 hours, reweigh the 2 sets of apparatus and observe the water levels in the conical flask. So, from this observation, we can see that an increase in wind speed increases the rate of transpiration. The movement of air carries away the water vapor from the stomata in the leaves, resulting in weight loss in the conical flask placed under the fan. In the next section, we will use the same apparatus and procedure, except that this time we will see how light affects the rate of transpiration. Here, the only additional apparatus is a spotlight. Place the conical flask with the balsam plant under the spotlight, and another in the dark cupboard in the same room. After 2 to 3 hours, reweigh both sets of apparatus and observe the changes. From this observation, we can conclude that light stimulates the opening of stomata and consequently increases the rate of transpiration. The higher the intensity of light, the higher the rate of transpiration. This is because plants carry out photosynthesis during daylight with open stomata. In our third section, we will evaluate how the rate of transpiration will be affected by the humidity of the air. We will use the same procedure as the previous experiments. Now, place the conical flask with the balsam plant inside a plastic bag containing dehydrated calcium chloride. After 2 to 3 hours, reweigh the two sets of apparatus and observe the water levels in the conical flask. In this experiment, we can see that the rate of water loss depends on the difference in concentration of water molecules in the leaf and in the atmosphere. High relative humidity reduces the rate of transpiration, thereby reducing the water loss. Factors like wind, light, temperature, and humidity affects the rate of evaporation and thus the rate of transpiration of water in plants.
Transpiration is very important to plants because of several reasons. Transpiration helps in the absorption and transportation of water and minerals in plants. The pulling force developed by transpiration provides the pathway through which water and minerals are transported in the plant. As water is lost or used up by the plant, more water is sucked up from the xylem vessels in the leaves, stem, and roots. The flow of water is continuous from the roots to the leaves. Plants get rid of their excess water through transpiration. By doing this, they can maintain the concentration of cell fluid. Evaporation from the leaf surface has a cooling effect which helps to prevent the heat of direct sunlight from damaging the delicate cells. During transpiration, the opening of the stomata allows carbon dioxide from the atmosphere to diffuse inside the leaf for photosynthesis, while oxygen diffuses out through the stomata into the air. So we have come to the end of this session. Let's recap what we have learned today. Number 1. The transport system in plants involve the xylem, which are dead cells that transport water and minerals from the roots through the stem to the leaves, where water vapor is lost through the stomata. This process is called transpiration. Number 2. The phloem, which is living tissues, transport glucose from photosynthesis in the leaves to the other parts of the plant. Number 3. The opening of the stomata enables the exchange of gases, that is carbon dioxide and oxygen, to diffuse in and out of the leaves. Number 4. The stomata also allows for the release of water vapor to the atmosphere. Number 5. Factors affecting the rate of transpiration include humidity, temperature, light intensity, and movement of air or wind. Number 6. Transpiration helps in the absorption of water and mineral salts by the roots. It helps the flow of water up to the leaves, and it also cools the plants. In closing, plants, like in humans, also possess their own unique transport system to move water, minerals and glucose to the rest of the plant to enable healthy growth.